Hello, everybody. Thank you for joining me today for this virtual seminar. My name is Haritha. I am a fourth-year graduate student in the Brambilla Lab, and today I'm going to be talking to you about one facet of my project, which is to study the role of TNFR2 signaling in neuroinflammation following CNS disease. As some of you may know, the Brambilla Lab is mainly focused. There we go. The Brambilla Lab is mainly focused on studying a disorder called multiple sclerosis. MS is a chronic neurodegenerative disorder that affects more than 2.5 million people worldwide. It is a T-cell mediated autoimmune demyelinating disorder in which the peripheral T cells infiltrate the CNS, bind to myelin fragments on antigen-presenting cells, which results in a release of pro-inflammatory cytokines and chemokines that results in acute inflammation, oligodendrocyte death, demyelination, axonal degeneration, and eventual neuronal loss. And MS patients exhibit a variety or can experience a variety of symptoms ranging from fatigue to vision problems to walking difficulty. And so it really, MS really does impair the quality of life in these patients. So most MS patients are originally diagnosed with relapsing remitting MS, which is characterized by periods of relapses where patients experience increased symptoms and or disabilities, followed by periods of remission where patients recover either completely or partially and plateau before another relapse, and so on. About 50% of these patients eventually develop into secondary progressive MS, which is characterized by this gradual worsening of symptoms and disability over time. The rarest form of the disease is called primary progressive MS, which is characterized by this gradual worsening of the disease from onset. And in this case, these patients do not experience any clear periods of relapses. So there's really two challenges in developing therapies for MS. One is to suppress the immune system or the inflammation that occurs in MS lesions. And the second is to actually promote remyelination and repair of the CNS. Current disease-modifying therapies for MS are immunosuppressants that work fairly well in slowing disease progression and decreasing the severity of these MS attacks. However, they are associated with severe side effects, and the main reason why we don't have more therapies for MS is because there's a baseline lack of understanding of the pathophysiology underlying MS. So we in the lab are interested in studying tumor necrosis factor, which has been implicated in MS pathophysiology. TNF is an immunomodulatory cytokine that regulates inflammation, cancer, autoimmunity, and infections. As you can see on the right here, there are two forms of TNF. There is a transmembrane TNF that can then be cleaved into the soluble form of the TNF. On the other side of this, there are two receptors. So there is TNF receptor 1 and TNF receptor 2. Due to differing binding affinities, soluble TNF preferentially binds to TNF receptor 1, and transmembrane TNF preferentially binds to TNF receptor 2. Depending on which receptor is activated or bound to, you get differing downstream signaling cascades. So TNF receptor 1 signaling is typically associated with apoptosis and inflammation, while TNF receptor 2 signaling is typically associated with cell survival and protective processes. MS patients have been shown to have increased levels of TNF in their serum and cerebrospinal fluid, as well as at the site of active lesions. And these TNF levels correlate with the severity of the lesions and the disease progression. So as you can see in the graphs below, the graph on the left is scoring the EDSS score, which is a disability score given to MS patients, with a higher score meaning worse symptoms. And as you can see, the higher the score, the more TNF levels they have in the cerebrospinal fluid. Similarly, the patients who progress faster along the clinical course of the disease also show to have higher levels of TNF, suggesting that tumor necrosis factor could be a therapeutic target for MS. So in order to study TNF signaling in MS pathophysiology further, investigators in the last few decades have turned to animal models. One of the most common animal models used to study MS is called experimental autoimmune encephalomyelitis. This is a model that is used to, or in this model, you induce BBB breakdown and an immune response to myelin-specific antigens, which then facilitates the infiltration and of peripheral immune cells into the CNS. And this results in an MS-like phenotype that can be scored on a scale from 0 to 6. So this is a good model to investigate the role of neuroinflammation in disease. And the other model that I'm going to be bringing up today is called the cuprizone model of demyelination, in which you administer this cuprizone diet to animals, and this results in rapid demyelination that is most evident in the corpus callosum. And this model of demyelination is a good model to study oligodendrocyte turnover and remyelination in the absence of a primary inflammatory response. So there are other models that are used to study MS symptoms and pathophysiology. However, these are the only two that I'm going to bring up today. So in the last 2 decades, there have been many studies looking into TNF signaling using the EAE and the cuprizone model. About 22 years ago now, I guess, one lab showed that by ablating TNF completely, it resulted in a worsening of EAE, suggesting that TNF signaling is necessary for, or is protective in EAE progression. However, when you look into the two receptors, you really find these differences where if you ablate TNFR1, you find these mice are resistant to EAE, while TNFR2 knockouts develop exacerbated EAE, suggesting that the presence of TNFR1 is detrimental, while the presence of TNFR2 is protective. Similarly, in the cuprizone model of demyelination, another lab has shown that ablating TNFR, or knocking out TNFR2 results in impaired remyelination following cuprizone-induced demyelination. Our own lab has published a study showing that pharmacological inhibition of soluble TNF signaling through TNFR1 is protective during EAE because of the presence of TNFR2 signaling still there. So these studies in combination have suggested that soluble TNF signaling via TNFR1 is detrimental, and transmembrane TNF signaling via TNFR2 is protective. And our lab is really interested in answering 2 questions. The first is, how does TNFR2 control or suppress the inflammatory response necessary for EAE development? And the second is, how does TNFR2 control or promote the remyelination capacity necessary for repair following injury? To answer these 2 questions, we as a lab have used a genetic approach in which we conditionally ablate TNFR2 from each of the cell types that it is expressed in the CNS. So TNFR2 is expressed in astrocytes, oligodendrocytes, OPCs, and microglia. Han Gao, a previous grad student in the lab, studied the role of TNFR2 signaling in microglia, and Placido Liano, a current postdoc in the lab, is looking into the role of TNFR2 signaling in astrocytes. For today's talk, I'm going to be focusing more on the oligodendrocytes and the effect of TNFR2 in these cells. So Pernille Madsen, a previous grad student in the lab, really laid down the groundwork for my project. She generated these CMP-free TNFR2 floxed mice in which TNFR2 is ablated from the whole oligodendrocyte lineage. So not only just the mature oligodendrocytes, but also the OPCs. And when she ablated TNFR2 from the whole lineage, she found that these mice have exacerbated clinical scores following EAE induction. So just as a reference point, on the x-axis, I have the days post-immunization. On the y-axis is the clinical score. A score of a 3 is considered full hind limb paralysis. A score of a 4 is considered full forelimb paralysis. So as you can see here, the black squares, which are the conditional knockouts, have increased disability or increased scores compared to the controls. Most of this difference in these two genotypes can be attributed to the chronic phase of the disease, which is typically associated with, or the clinical scores during this period are typically associated with mechanisms of repair and remyelination that are occurring or not occurring during this time point. So Pernille looked further at this chronic phase, and she found that TNFR2 ablation in oligodendrocytes resulted in increased oligodendrocyte loss following EAE induction in the chronic phase. And when she went in vitro and conducted a differentiation assay, she found that TNFR2 TNFR2 ablated OPCs have impaired differentiation into mature oligodendrocytes in vitro. So her study that was published back in 2016 showed that TNFR2 signaling in OPCs is necessary for oligodendrocyte differentiation. In addition to the chronic phase of the disease, we also observed in these mice an exacerbation of the clinical scores during the acute phase, suggesting that TNFR2 signaling in oligodendrocytes is protective during acute EAE. And this acute phase of the disease is, rather than being associated with reparative processes, such as like in the chronic phase, this acute phase, or the clinical scores during this acute phase are more associated with the neuroinflammatory cascades that are occurring due to the infiltration of peripheral immune cells into the CNS. So we decided to look further at this phase, or at these time points, to see if indeed oligodendroglial TNFR2 could be playing a role in the infiltration of these cells. And indeed, we found that in the conditional knockouts at the acute pre-symptomatic as well as acute phase of the disease, we found that there's an increased infiltration of CD3 positive T cells into the spinal cord, as well as increased presence of CD45 positive monocytes and IVO1 positive microglia in these cells, or in the spinal cord. So this study, in addition, we found that by ablating oligodendroglial TNFR2, there's an acceleration of the expression of these pro-inflammatory molecules that are typically expressed following EAE. As you can see, we see increased expression of TNF, interferon gamma, TNFR1, IL-1 beta, IL-6, IL-2, CXCL1, and so on. So in order to determine if this was an indirect effect of ablating TNFR2, or whether oligodendrocytes themselves are directly influencing this inflammatory response, we again went in vitro and conducted an assay, and we stimulated mature oligodendrocytes with a cocktail of TNF, interferon gamma, and IL-1 beta, and found that indeed mature oligodendrocytes can produce inflammatory cytokines. They massively upregulate the production of multiple cytokines following stimulation. However, this response is not TNFR2 dependent. On the other hand, in OPCs, OPCs also do increase response or increase production of inflammatory molecules following stimulation. However, or not however, but some of these molecules that we probed for were indeed TNFR2 dependent, suggesting that TNFR2 signaling in OPCs is not only necessary for oligodendrocyte differentiation, but also it suppresses the inflammatory response following stimulation with inflammatory molecules. Of course, these two conclusions are based on in vitro data. So my project has been looking into whether or not this is still the case in vivo. And in order to study TNFR2 signaling in OPCs, I could not use Pernille's mouse model with the CMP pre-line, because CMP is expressed throughout the whole lineage, from the OPCs all the way through to the mature oligodendrocytes. So when I first joined the lab, we generated a transgenic mouse model called the PDGFR alpha CRE TNFR2 Flox EYFP mouse line, in which the CRE is under the transcriptional control of a PDGFR alpha promoter, which is selectively expressed in OPCs in the CNS. And upon tamoxifen administration, we can induce the ablation of TNFR2 and the simultaneous expression of EYFP, allowing us to fate map these OPCs in the mouse. In addition to the knockout mice, we also generated a control colony in which the administration of tamoxifen does not induce the ablation of TNFR2, but it still induces the expression of EYFP. So because this is a novel mouse model that our lab has generated, we first have to characterize these mice. I'm not going to go through the specific data today. I'm just going to quickly summarize it before moving on. But in terms of recombination efficiency, we found that about 70% of these PDGFR alpha positive cells also express EYFP following tamoxifen administration. And in this PDGFR alpha positive EYFP positive population, there's about a 50% ablation of TNFR2. Also, just as a full disclosure, PDGFR alpha has been shown to also be expressed in fibroblasts. So I conducted a flow cytometry experiment to see how many of my PDGFR alpha positive cells in the spinal cord also express PDGFR beta. And it turns out the number or the percentage is fairly small. It's only about 1% of the PDGFR alpha positive cells that are also PDGFR beta positive, suggesting that that small 1% are in fact fibroblasts. So moving on, I also conducted behavioral experiments under naive conditions to see if there's any basal deficits or differences in these mice. And I found no changes in the locomotor behavior or the locomotor coordination using the open field and rotor-rod tests. So as I said before, my interest is, or my project focuses on looking at TNFR2 signaling OPCs in both neuroinflammation and remyelination. So to study neuroinflammation, I am using the EAE model with the females that I get from my mouse colonies. And then for the Cooper zone, or I'm studying remyelination using the Cooper zone model and the males that I generate in my mice colony. For today, in the interest of time, I'm only going to be discussing the data I've generated with the EAE model. So as I mentioned before, this is a model in which we inject a myelin peptide sequence and pertussis toxin to facilitate the infiltration of peripheral immune cells into the CNS, and this results in an MS-like phenotype. So when I induced EAE in my mice, I found that when TNFR2 is ablated in the OPCs, there is an earlier onset and peak disease following EAE induction, suggesting that OPCs in fact do play a role in this acute phase of the disease, which is typically associated with neuroinflammation. So in order to really to answer the question of whether TNFR2 ablation in OPCs affects the infiltration and presence of the immune cell populations in the CNS, I conducted a flow cytometry experiment in the spinal cord at pre-symptomatic, so 12 dpi, acute at 18 dpi, and subacute at 25 dpi, to probe for different immune cell populations that are known to infiltrate the CNS following EAE induction. And so, as you can see here, we found that TNFR2 ablation in OPCs accelerates CD4 positive T cell infiltration, but not CD8 T cell infiltration into the spinal cord. We also observe no changes in the presence of B cells in the spinal cord, which are barely there to begin with. And there is also a significant acceleration of the neutrophil and natural killer cell populations at the acute phase of this disease. In addition, there's no changes in the actual infiltration of macrophages into the spinal cord. However, there is a significant increase in the activated stage of these macrophages in the spinal cord, which we measured using MHC class 2. When we looked at the resident immune cells of the CNS, the microglia, we found that there is an increased microglia number, as well as increased microglia activation in the spinal cord following EAE induction. So in summary, what I've shown you so far is that TNFR2 ablation in OPCs results in earlier onset and earlier peak disease following EAE induction. And it seems that TNFR2 ablation in OPCs results in the accelerated infiltration of CD4 positive T cells and neutrophils, as well as increased activation of macrophages and increased number in microglia. So there's still a lot to be done for this. Some future experiments I have planned is to do a qPCR of RNA that's extracted from YFP sorted cells during pre-symptomatic EAE to identify genes that are regulated by TNFR2. In addition, I plan to do a multiplex analysis of inflammatory mediators during the acute stage of EAE, as well as a histological analysis of demyelination at these various stages of EAE to see if TNFR2 ablation in the OPCs could be affecting demyelination or remyelination as well at this time point. So, so really the conclusion that I've shown you so far from the in vivo data is that TNFR2 signaling in OPCs does suppress neuroinflammation during EAE. But the question here is, what is the underlying mechanism? So to determine what this mechanism is, I decided to conduct an RNA sequencing experiment on in vitro primary cultured OPCs from wild type and TNFR2 knockout pups. And so I isolated these cells using the Miltenyi magnetic bead system. And I either cultured them for 3 days under non-stimulated condition in their basic media with PDGF and FGF, or I cultured them for 3 days and then stimulated for 3 hours with TNF, interferon gamma, and IL-1 beta for 3 hours before collecting the RNA. And this RNA was sent to the Beijing Genomics Institute for them to conduct or to complete the RNA sequencing. And then I received the raw files, which I then aligned to the mouse genome with STAR, and then conducted the differential gene expression analysis using EDGE-R. So just to make it a little bit more clear, the samples that I submitted there were 4 groups. So there's the wild type OPCs, non-stimulated wild type OPCs that were stimulated, the TNFR2 knockout OPCs, non-stimulated, and the TNFR2 knockout OPCs that were stimulated. And when I compare all of these groups to each other, we find thousands of genes that are differentially regulated, either up or down, dependent on stimulation versus non-stimulation, or dependent on TNFR2 presence or lack thereof. So in order to filter through this data set, because it is a lot of genes and it can get very overwhelming very fast, I specifically isolated different combinations of genes. So the first question I wanted to look into was whether or not TNFR2 ablation in OPCs exacerbates their response to neuroinflammatory stimuli. So I created a Venn diagram of the different genes that are differentially expressed within each of my comparisons. So I know it's a lot of numbers and figures, so I'm going to go through it really slowly. So the pink here is comparing the TNFR2 knockout stimulated versus the wild type stimulated OPCs, while the blue is the TNFR2 knockout non-stimulated cells versus the wild type non-stimulated. And the gray is wild type stimulated cells versus the basal non-stimulated wild type OPCs. And the yellow here is the TNFR2 knockout stimulated cells versus the TNFR2 non-stimulated cells. As you can see, a majority of these genes are actually similarly upregulated or downregulated between following stimulation, and they're not dependent on whether or not TNFR2 is present. However, the group that I was really interested in to look at exacerbation of TNFR2 dependent exacerbation of genes following stimulation is this group that I circled or selected here in a square. And so just to explain it a little clearer, so here I have a little plot explaining how these comparisons work together. So for the number that I squared or I selected, in the wild type cells, there is either an upregulation or downregulation of those genes following stimulation. And in the knockout cells, there is also either an upregulation or downregulation of those genes following stimulation. However, the real big difference here is not only is this either upregulated or downregulated in both genotypes, but that effect is exacerbated significantly in the knockouts compared to the wild type stimulated. And when I did a gene ontology analysis of just those 350 genes, I found that most of the upregulated genes are involved in the inflammatory response, while most of the downregulated genes are involved in cell cycle and cell division. So these were two pathways of interest that I wanted to follow up on, in addition to looking at what genes are exacerbated or following stimulation in a TNFR2 dependent manner. I also wanted to see what processes are selectively TNFR2 dependent in non-stimulated and stimulated OPCs. So in the stimulated OPCs, there's about 284 genes that are selectively upregulated in TNFR2 knockout stimulated OPCs compared to wild type, and there's about 450 genes that are significantly downregulated. These genes are associated, the downregulated genes are more associated with cell cycle and cell division, while the upregulated genes are more associated, it seems, with lipid metabolisms, DNA damage, and oxidation reduction processes. So, and then looking at the genes that are significant or selectively up or downregulated in the non-stimulated TNFR2 knockout OPCs, found again that the metabolic processes seem to be the most upregulated due to the ablation of TNFR2. So in addition to the immune system response and the cell cycle and cell division, I also wanted to look into the metabolic processes that are affected due to TNFR2 ablation in these cells. So first, I'm going to go through the immune system response, or the genes that are associated with the immune system response. So TNFR2, or stimulating OPCs with this triple cocktail of TNF, interferon gamma, and IL-1 beta results in an upregulation of genes that regulate microglia proliferation. So CSF1, CX3CL1, IL-33, they're all genes that are known to promote microglia proliferation, and IL-33 in particular seems to be more increased to a higher significant amount in a TNFR2 OPC specifically following stimulation. In addition to microglia proliferation, the stimulated OPCs also upregulate genes that promote T cell migration in a TNFR2 dependent manner. So as you can see here, I've shown you a list of cytokines and chemokines that are not only do wild type OPCs upregulate the expression of these molecules, but they also, the TNFR2 knockout OPCs seem to upregulate them even more, like CCL2, CXCL10, CXCL12, and CXCL16. And so This, in summary, shows that wild-type OPCs can produce cytokines and chemokines when exposed to a neuroinflammatory environment. And TNFR2 ablation in OPCs increases the expression of these cytokines and chemokines further compared to wild-type OPCs following stimulation. And this data also correlates nicely with the in vivo data I showed you before, in which we see through flow cytometry an increased infiltration of T cells and as well as increased microglia number during acute EAE in the knockout animal. So for future experiments, I'm planning on conducting a multiplex analysis of the media that these cells were cultured in to see what inflammatory mediators are actually secreted by these OPCs. And then I am also planning on doing a qPCR of RNA extracted from YFP sorted cells at the pre-symptomatic time point to validate these gene expression changes that I see in vitro and see if these also correlate to gene expression changes that occur in vivo. So moving on, the next pathway of interest that I wanted to look into is the cell cycle or cell division processes. So OPCs are highly proliferative cells, and so two specific pathways that I wanted to look into is OPC proliferation as well as OPC differentiation. So in general, OPC proliferation genes and OPC markers are downregulated following stimulation in both the wild type and the TNFR2 knockouts. Some, but not many, are TNFR2 dependent. PDGFR alpha, for example, is significantly downregulated in the TNFR2 knockouts following stimulation. And LIN is another gene that is significantly downregulated here as well. So, yeah. On the other hand, genes associated with OPC differentiation are also differentially regulated in a TNFR2 dependent manner. And however, so on the top two rows of genes I have here are positive regulators of differentiation, while the bottom row here is negative regulators of differentiation. As you can see, most of the positive regulators of differentiation are downregulated in both the wild type and TNFR2 knockout OPCs. Some are TNFR2 dependent, some are not, and corresponding to the downregulation of positive regulators, there's also an upregulation of the negative regulators, suggesting that differentiation is impaired, or the differentiation process is further halted because of increased presence of inhibitory factors in these OPCs. So in order to see if this was actually the case, I conducted an in vitro experiment where I isolated wild type and TNFR2 knockout OPCs, cultured them 3 days with PDGF and FGF, and then either did not stimulate them or stimulated with 3 hours with the same 3 molecules, and then cultured them for 4 days in a differentiation media that contains T3 and CNTF to see if differentiation of these cells is altered in a TNFR2 dependent manner. And what we found was that OPC differentiation decreases following stimulation. So on the graph on the left here is showing the percent of O4 positive cells out of the total OLIG2 population. So this cell population includes the immature oligodendrocytes or intermediate oligodendrocytes as well as the mature oligodendrocytes. And then the graph on the right here is showing the percent of MVP positive cells as a total out of the total O4 positive population. So this is selectively the mature oligodendrocytes that are capable of producing myelin. So as you can see here, in basal conditions, so non-stimulated, the TNFR2 knockout OPCs do have a decreased differentiation into O4 positive cells compared to wild types. However, following stimulation, there is no difference between the two genotypes. So in summary, what I've shown here is that OPC proliferation and OPC markers are downregulated following stimulation. and genes that are associated with OPC differentiation are differentially regulated in a TNFR2 dependent manner. However, there is no difference in the actual number of cells or OPCs that differentiate into mature oligodendrocytes following stimulation, suggesting that it may not be an impaired differentiation, but rather maybe an impaired capacity to myelinate or produce proteins involved in myelination. So some future experiments that I have planned is to conduct a morphological analysis of these differentiated oligodendrocytes in vitro to see if there may be, in fact, morphological differences that are TNFR2 dependent. And in addition, I will be validating some of these genes with qPCR from RNA extracted from differentiated oligodendrocytes to assess if there are TNFR2 dependent differences in myelin-associated genes at that time point. So the last pathway of interest to me is metabolic processes. So the metabolic pathways are highly complex. There are hundreds of intermediate proteins and genes that are involved in maintaining homeostasis in a cell, and it can get very, very complicated very fast. But the fact that metabolic processes showed up as such a big response, a TNFR2 dependent change, suggested that maybe the TNFR2 ablated OPCs have some sort of metabolic stress that is occurring. And indeed, we found that TNFR2 knockout OPCs upregulate genes that promote the generation of reactive oxygen species. So NOS2, SOD2, and SOD3 all promote the production of reactive oxygen species, which are known to activate stress-related pathways and cause metabolic changes. One such pathway that I'll be focusing on for this talk is the NRF2 KIP1 signaling pathway. So just very briefly, when oxidative stress occurs, typically through the presence of reactive oxygen species, NRF2 and KIP1 are typically bound together under basal conditions, resulting in proteasomal degradation. In the presence of stress, KIP1 is dissociated from NRF2, resulting in translocation of NRF2 to the nucleus, and where it then binds to the antioxidant response element, resulting in the gene expression or the upregulation and transcription of genes associated with antioxidant response. So NRF2 has been shown to regulate multitude of pathways from maintaining cellular homeostasis to lipid metabolism and lipogenesis, as well as NADPH generation and synthesis and apoptosis. As I was going through the literature, this pathway became more and more interesting to our lab to look into further to see if TNFR2 signaling could be, or if NRF2 could be downstream of TNFR2 signaling. And we found that NRF2, the gene that encodes for NRF2, is differentially regulated in OPCs in a TNFR2 dependent manner. So TNFR2 ablated OPCs have increased expression of NRF2, suggesting that there is some sort of response to the stress that we see, or that correlates with the presence of increased genes associated with the production of reactive oxygen species. In addition to increased expression of NRF2, there's also increased expression of antioxidant-related genes that are downstream of NRF2. HMOX1, GCLM, and AKR1B10 are all upregulated following stimulation, and two out of these three genes is upregulated in a TNFR2 dependent manner. So, in addition to the antioxidant response, NRF2 also plays a role in lipid and glucose metabolism. So I looked into the specific genes that are downstream of NRF2 for lipid and glucose metabolism, and found that these genes are also differentially expressed in OPCs following stimulation. And in fact, most of these genes are downregulated in the OPCs following stimulation, which could be could correlate to the decrease in the proliferative genes as well as the differentiation genes that I showed you before, because without proper lipid and glucose metabolism, it is difficult for OPCs to actually undergo cell proliferation and differentiation. So in summary, what I've shown you here is that TNFR2 ablation in basal OPCs may result in increased metabolic stress, and TNFR2 ablation in OPCs following stimulation further exacerbates the metabolic stress, specifically in relation to glucose and lipid metabolism. And these genes that are differentially expressed are downstream of NRF2 signaling, with an increased expression of antioxidant genes and a decreased expression of genes associated with lipid and glucose metabolism. So some future experiments I have planned. We have a collaborator in Denmark who has sent us several KIP1 inhibitors in vitro, and I am planning on testing these inhibitors to see if the gene expression changes that are observed in TNFR2 ablated OPCs can be reversed. And in addition, I'm planning on conducting a transient transfection of OPCs with the RE2P vector provided by Promega to measure TNFR2 dependent NRF2 activation, which if there is this correlation, it would directly link TNFR2 signaling with NRF2, with the NRF2 pathway. So to sort of summarize everything that I've shown you today, as you all know, wild-type OPCs typically go through two processes, both in vitro and in vivo. They can either proliferate and generate more OPCs, or they differentiate into mature oligodendrocytes. However, in neuroinflammatory environment, what we found is that wild-type OPCs typically decrease their differentiation and proliferation, but instead increase the production of inflammatory cytokines and chemokines, thereby contributing to the neuroinflammatory environment that already exists. On the other hand, in TNFR2 knockout OPCs, they are already seem to be under some sort of metabolic stress, which results in a slightly impaired differentiation into mature oligodendrocytes. And this is further decreased when these TNFR2 ablated OPCs are in a neuroinflammatory environment. So they have impaired differentiation or decreased differentiation, decreased proliferation, but also massively increased production of cytokines and chemokines that is typically suppressed by the production or the presence of TNFR2. So finally, I would like to thank everyone in my lab. Roberta, my mentor, it's been great. She really provides great insight into my project. Placido, who's the postdoc in our lab, Melanie, fellow grad student, as well as Eva and Shafayat, who take care of our mouse colonies. I'd also like to thank my thesis committee for all of their input into my project, as well as Jan at the Miami Project Imaging Corps for all of her help with the stereology, as well as the Solomics. And Oliver at the Flow Cytometry Corps. He is an amazing resource. And then I also do want to acknowledge the Lambertson Lab for providing us with, or for allowing us to use their plates to conduct our mesoscale analysis, as well as the Bach Lab, who sent us their KIP1 inhibitors to test for our own assays. In addition, Matt Danzy, who's a previous grad student in Vance's lab, really, he taught me everything I know for how to analyze RNA sequencing data sets, so he deserves a huge shout out. and of course the Beijing Genomics Institute for actually conducting the sequencing. And yeah, I think that's it. I will be happy to take any questions now.
All right. Well, if you have a question, feel free to just let me know on the bottom. You can even click the yes, and I'll go ahead and unmute your mic, and you can ask your question. Okay. It appears we don't have any questions, so we'll go ahead and end for today. Remember, we have another scheduled lecture next Wednesday, and I'll be updating the announcements as other departments add to those. Again, I'll give you another minute or two if somebody else has another question. If not, I'll go ahead and just end the meeting. And Haritha, thank you for participating. Oh, okay. I'll go ahead and unmute Dr. Dietrich. One moment, please.
Okay.
Hi, Maria. I think there's some questions on the chat box.
Let's see.
John had a question and Nagi did.
Okay. See, I don't see them on my end, but
They
I'll go
were sent
ahead.
to everyone.
Oh, I see it.
You see it, Haritha?
Yeah, from Nagi. Yeah, he was asking if I looked at protein levels for, especially for ID2 and NRF2. So I have not. So these, that is the next step in this process. I have the protein collected, just a matter of running the Western blots. And I will say that at least for ID2, I have done a Western blot before in just basal conditions, not following stimulation in the OPCs. And I do see an increased expression of ID2 in the TNFR2 knockouts. Yes, they are. Yeah.
Haritha, this is Jay. Can I ask a question?
Yeah.
So you stimulated in your in vitro assays, you stimulated with TNF. So this would be soluble TNF?
Yes, this is soluble TNF.
So can you comment on how stimulating soluble TNF might influence or might be affected by TNFR2 deletion, which is presumably stimulated by transmembrane TNF?
Yeah, so I will say that when I stimulated these cells, they were about 80% confluent. So many of these OPCs or the processes were in contact with each other, and OPCs have been shown to produce transmembrane TNF. So it's not that transmembrane TNF with TNFR2 signaling is not occurring, but the reason we stimulate with soluble TNF is mainly because that is what is available to us. As far as I know, there is no, or the actual protein structure for transmembrane TNF is not really sold to us as a recombinant protein. And typically TNFR1 and TNFR2 sort of have are dependent on each other. So TNFR1 typically is detrimental while TNFR2 is protective. So when there is a lack of TNFR2 signaling, there is sort of this tipping of the scales towards TNFR1 signaling, which could be why we see increased inflammatory response in these OPCs.
Hi, this is Dalton. Great talk. Thank you so very much. Some really interesting cell biology and mechanistic questions. And I think you answered it a little bit at the very end, which was my question, which I ended up erasing. And that had to do with translational potential for this targeted therapeutic intervention for these patients that are suffering from these demyelinating disorders. So can you just expand on some of those compounds or drugs that you're going to start working with this company to maybe move into a more translational program?
Yeah. So so I should have gone into more detail, but so the NRF2 pathway has been extensively studied, especially in relation to cancer biology. And as I mentioned before, usually under basal conditions, KIP1 binds to NRF2, which results in proteasomal degradation. And in response to stress, this protein complex is dissociated, allowing NRF2 to translocate to the nucleus. Usually, theoretically, if we want to promote the production of NRF2 or the translocation of NRF2 to the nucleus, resulting in more antioxidant genes expressed, which would theoretically be protective, the easiest way to do that would be to inhibit KIP1 binding to NRF2. So this lab in Denmark has generated several inhibitors, reversible inhibitors of KIP1 that bind to different binding sites that can associate KIP1 and NRF2 typically. And theoretically, what this should do is it'll sequester the KIP1 away from NRF2, which results in more translocation into the nucleus and more production of these antioxidant genes. Another thing that I did not mention here is that NRF2 has also been shown to suppress the production of inflammatory molecules. So typically when you activate NRF2 signaling, you also see decreased expression of inflammatory molecules such as CCL2, IL-1 beta, and other chemokines and cytokines. So Nagi just asked, do these compounds have to cross the BBB? The lab in Denmark has done some in silico assays to see whether or not these compounds are capable of crossing the blood-brain barrier, and everything that they've sent us so far seems has a good score on that scale. However, the first step for us to see is to see whether or not these compounds are actually able to do anything in vitro.
Great. Thank you very much. That's great. Yeah, that opens up a whole new target for different types of demyelinating disorders, a possibility, or other neurodegenerative diseases. So it's great that you've got a collaboration that's going to be working on this.
Yeah.
Haritha, check messages. You have a message to a question there.
Oh.
Yeah. Roberta's giving you a message.
Yes. So so we received these compounds a couple months ago, and or not months, but a couple weeks ago, and because OPCs are a little harder to culture and generate, I decided to first test these compounds in immortalized microglia line. And so we received about 10 compounds from their lab, and I have tested so far 7 or 8 of them just one replicate. And we have found that several of these genes, some of these compounds, interestingly, are able to increase the expression of antioxidant genes, while others don't increase the expression of those genes, but in fact, instead suppress the expression of inflammatory molecules such as CCL2 and IL-1 beta. So these compounds do work in vitro, at least for the immortalized microglia line. The next step that I was planning on doing before this whole self-isolation situation was to test them in vitro. Hopefully, sometime during the summer, I should be able to test these compounds out in my OPCs. Let's see. I think that's everything, Maria.
Okay. I'm sorry about that. I couldn't see the other questions, but those
I think there is a question from Shyam on microglia. The first question. I don't think you addressed that.
I don't see.
Sham was asking any analysis on microglia.
About what does this pertain to?
Not sure. Possibly he was referring to the mice, the knockout mice.
Any analysis in the spinal cord on microglia? Okay. Yeah, so we do, I did do the flow cytometry and just preliminary immunofluorescence of the spinal cord tissue at the pre-symptomatic and acute phase, and we do see increased presence of microglia in the spinal cord. I have also isolated, FACS sorted out the microglia at the pre-symptomatic phase of EAE in both my, or I'm in the process of isolating these cells out in both the knockouts and the controls to see if there's any indirect effects or if it's possible that the OPCs are interacting with the microglia in the spinal cord.